Porgy, which was published last year in the peer-reviewed journal Marine and Coastal Fisheries. And I also want to acknowledge my co-authors, Wally, Dawn, and Marcel, as well as the numerous number of folks that have collected data for this project for the last 30 years. So it's a huge team effort, and it involves a lot of people, a lot of moving parts to put this sort of data set together. So I really appreciate that. And I also appreciate everybody listening in today. I know some people are in because of weather, and that's really unfortunate, but hopefully next week will look a little bit better as time goes on. All right, so Chip gave a little bit of an overview on Red Porgy in the South Atlantic, but wanted to make sure everybody's kind of on the same page to start with. So the Red Porgy stock off the Southeast U.S. Atlantic coast, or the South Atlantic, was initially assessed through the Southeast Data Assessment and Review, or SEDAR process in 2001, after a lot of stakeholders, which included fishers, scientists, other participants in the region, expressed concern that their numbers seemed to be dwindling. That assessment in 2001 found that they were indeed overfished and overfishing was occurring. And this initial assessment in 2001 led to an 18-month-long moratorium, followed by some pretty strict involved regulations on catch, and has been in a rebuilding phase for a long, long time. So there have been a few updates to this assessment that continue to find the same overfishing and overfish status over the years. And in 2020, the most recent stock assessment was completed through the SEDAR process with commercial, recreational, and fishery independent data through 2017. This new assessment included updates to data sources and methods that had advanced since the original SEDAR assessment in 2001. Unfortunately, though, the 2020 assessment came back to the same conclusions as the previous ones, categorizing South Atlantic Red Porgy as overfished and with overfishing occurring, despite regulations and despite the rebuilding plan. On your screen is a plot of this ratio of spawning stock biomass to minimum standing stock threshold from this assessment. And the ultimate goal of these plots is to show where we are relative to that one line. And the goal is to be above the line. But unfortunately, South Atlantic Red Porgy have barely even approached that line in almost 30 years, despite management efforts. On the left are the stock projections through 2026 from that same assessment in 2020. And the goal here is to reach the blue and green lines. However, the projections, such as this one that was derived from fixed fishing mortality that provides a P-star of 50%, showed that unless something really drastic changes, the likelihood of the stock recovering to these management goals at those green and blue lines in the next 10 years or so were relatively small, or even extremely small. And during the course of the assessment, a couple of potential issues with the stock were identified that may help to explain its failure to recover. This included a pretty notable scarcity of new recruits to the population, as seen by the reduction of the red bars here in the population by age plot. In addition, the oldest individuals were also scarce, which is shown by the almost lack of blue and purple bars in the most recent years, to the point where you really can't even see the colors. In the case of Red Porgy and many of the other federally managed species in our region, the lack of older fish is an issue because they're protogynous hermaphrodites, meaning that younger fish are female and fish transition to males as they get older and larger. And the lack of these older males and, based on the few red bars, the younger females, puts the population at risk for limitations to spawning. The lack of new young fish also means that there are no replacements for fish being removed from the population. And so there seem to be issues on either end of the age spectrum. And the third item to note from the assessment was the potential for discard mortality. Discards are driven by things like low quotas, minimum size limits, market pressures, low bag or trip limits, and these things together ultimately mean that caught fish are more likely to be released because they hit one of these restrictions or some sort of desirability phase. And for Red Porgy, post-release mortality ranged from 27 to 64%, depending on where and how they were captured, what sizes they were, and if things like barotrauma was mitigated or not. So meaning that, in this assessment, there was somewhere between a 1 in 4 to a 2 in 3 chance that discarded fish aren't going to contribute to the future of the stock. As Chip mentioned, one of my roles here at South Carolina Department of Natural Resources is working with the Southeast Reef Fish Survey, which is a region-wide offshore fishery independent survey conducted as a collaborative effort through the Marine Resources Monitoring, Assessment, and Prediction Program, or MARMAP, and the Southeast Area Monitoring and Assessment Program, South Atlantic, or SEAMAP. Both are housed here at SCDNR, and we also have a federal partner for this program at the Southeast Fisheries Independent Survey, which is housed at the Southeast Fisheries Science Center in Beaufort, North Carolina. The primary gear used by SERFS and most applicable to Red Porgy studies is the chevron trap shown here on the left. MARMAP started the trap survey in 1990, and the other partners joined over the last decade. We currently deploy about 1,500 chevron traps on randomly selected stations between Port St. Lucie, Florida, and Cape Hatteras, North Carolina each year. Red porgy are one of the more common species encountered, or at least they were historically, and data recorded by the survey includes abundance or number per trap of each species caught, lengths of each species caught, and for some species, in particular red porgy, we dissect fish to collect otoliths for aging. Now, since the trap survey encompasses a very long time series, things like funding, scope of work, and the additional participation of SEAMAP and SEFIS have changed the spatial footprint of the survey over time, with what has become a much more even distribution of sampling across the region in more recent years. So for example, at the top left panel shows the beginning of the MARMAP chevron trap survey in 1990. And for the most part, sampling was pretty concentrated off of Georgia and South Carolina until about 1996. And in this year, the survey really started to expand more consistently into Florida and North Carolina. And that pattern of MARMAP expanding continued up to about 2009 before the transition between MARMAP only and the full SERFS complement. And the final panel, 2015, which is just sort of a representative year, shows the full SERFS region-wide effort. And all those black dots represent individual trap deployments. Traps since 2010 have been equipped with video cameras, which record, among other things, habitat type at trap stations. The chevron trap survey focuses on sampling hard or live bottom habitats, which includes a variety of bottom types containing some combination of rock and attached algae or invertebrates. With the most common invertebrates that we see are things like sponges, soft corals, sea whips. And these can range from fairly flat pavements that often have some type of sand veneer to larger 10, 15-foot ledges that are still trappable. And one of the other things that we record with each trap deployment is bottom water temperature as a habitat descriptor. So because of the experience that many folks around here had had with this long time series, we already had a pretty decent guess on what the stock assessment was going to say, especially about reduced numbers and few recruits. But we wanted to examine the population more specifically to see if both recruits and the spawners were undergoing the same patterns of decreased abundance in the region, using both the MARMAP historical data and the contemporary SERFS data. We also set out to define the spatial distribution for both life stages of red porgy in the region, as well as to define each stage's habitat use. And finally, we wanted to see if either the spatial distribution or the habitat use had changed over time. And the overall hope for this work was that it would give us a better understanding of what areas or habitats would be preferred or essential for red porgy to in turn better guide potential management approaches. There's been a lot of work done by biologists looking at why animals that are pretty mobile are in the places that they are, and a lot of the work comes down to opportunity. Mobile animals tend to occur in areas where they have good opportunities for finding food, for finding mates, for not becoming food, and for being either in livable conditions or maybe even good conditions for growth and survivorship. Things like being in the right temperature range, the right amount of sunlight, the right salinity, whatever that thing may be. I remember this very distinctly from when I was an undergraduate. So a really common way to show this to students when they're sort of early in their biology careers is to take them to feed ducks in a pond. When you arrive at the pond, the ducks are generally pretty spread out. But once you set up feeding stations, with some sort of bird seed or what have you, they become more concentrated where there is access to food. And you can see them changing that distribution because of that opportunity. Several years ago, Joey Ballinger, another scientist here at SCDNR, gave a really great analogy in the Council's newsletter about how football fans are really similar to fish, at least in some ways. One example of this is that in an uncrowded stadium, fans will concentrate at the places with the best views. For example, something like the 50-yard line, the end zones, whatever their preference may be. But once it gets crowded, more and more people have to settle for areas with less optimal views, like the nosebleed section or behind poles and dividers and things like that. And that same concept applies to fish since they're mobile. They can move to optimal areas or conditions, at least to a point, and if those aren't available, they can then utilize less optimal or marginal areas or conditions. And overlaid on top of these sort of basic biological concepts of being able to move in preferred areas or habitats. So those sort of set up the story for what potential changes you may see in distribution of fish. But in addition to that are the potential impacts of changing environments in any given region. I worked up in Alaska for several years before coming here to South Carolina. And up in the Bering Sea, there was a lot of concern that in that area, as temperatures warmed and ice coverage decreased, fish were moving with their preferred temperature range into the Bering Sea and potentially disrupting the ecosystem. Arrowtooth flounder was one of these more southerly species that like slightly warmer waters, all of 40 degrees Fahrenheit, that seem to be following this pattern. So the left panel here shows the historical distribution of arrowtooth flounder, where red is high biomass area, blue is low biomass to almost no arrowtooth flounder. And this is from 1983 from a trawl survey that's been conducted throughout the years over in the Bering. So for the most part, historically, arrowtooth flounder were really distributed offshore, more to the southern end near the Aleutian Islands. But by 2018, arrowtooth flounder had moved inshore, they had moved north, and they'd become much more common in that region. So in this case, the movement of arrowtooth flounder was really concerning because they aren't a harvested species or one of any major commercial use because the meat is pretty low quality. So it's affecting the economics up there. And in addition, they also eat young walleye pollock, which are a huge industry up there. So that combination of the habitat preferences for fish and the potential for a changing system and the ability of fish to move around are the context we were thinking of when we started this work with Red Porgy. So the data used for this project, we focused on the trap time series from 1996 to 2019 because of that more consistent coverage of Florida and North Carolina compared to earlier years that I mentioned. One of the advantages of using a long-term fishery independent survey like this one to look for changes is that we've maintained the same practices for the whole time series, and our work is unaffected by management regulations. Data used for analyses included the abundance and ages from fish collected in traps deployed as part of our standardized sampling. In some cases, in very few cases, length was used as a proxy for age if age was unavailable for some reason. Abundance per trap was split by the percent of red porgy in each life stage, which was defined as any age 0 or 1 fish were considered recruits, and anything older than age 1 were adults. So in other words, the fish we expected to be our spawners. David Wyansky here in our lab has been working on this for a while, but there does seem to be some variability in the age at which spawning becomes most prevalent in red porgy. But age 2 is about the time that spawning becomes pretty common in red porgy, so that's where our cutoff lied. In addition to the data collected during sampling, such as location, depth, and temperature, we also included 3 additional indicators of habitat type derived from the cameras for each station sampled. These were biota density, hard substrate coverage, and relative vertical relief. So the biota density is a continuous variable. It ranges from a value of 0% to 100%, where 0% means that video footage found no algae or invertebrates attached to the bottom, and 100% means that all visible bottom was covered with some sort of attached algae or invertebrates. Hard substrate coverage worked in a very similar way to biota density, ranging from a value of 0% to 100%, where 0 means that video footage found no rocks, no boulders, or any type of ledge, and 100% means that all of the visible bottom was some sort of hard rock. And vertical relief was a relative classification of low, medium, or high, where low areas were something like a pavement and high areas were distinct ledges. All right, to analyze the data, we used a method called the Vector Autoregressive Spatial Temporal Model, or VAST for the rest of this. I'm not going to repeat it again, to standardize abundances of each life stage over time and to define their spatial and depth distributions. If you sat in on one of the recent stock assessments, this type of standardization is very similar to what has been done in recent years for indices of abundance from either surveys or logbook data or landings data. And the VAST model works through a 2-step generalized linear model that basically looks at the likelihood of a red porgy being in a trap, and if they are in that trap, how many of them are there, and it effectively multiplies those 2 values together. We also were able to include those other things like habitat type and temperature that we collect for each trap that I mentioned earlier into a single model that effectively corrects the abundance for all of those things. VAST models also deal pretty well with 0 catches, which are common in fishery independent surveys that are not targeted to a single species. And the VAST model also deals with the fact that it's very likely that traps near each other have similar red porgy catches, which is something called spatial autocorrelation. In addition to VAST, we also used a more focused generalized linear model with interactions between abundance and habitat and year to test whether or not there have been any changes between red porgy abundance and what habitats they're using, and if that has changed over time. In this case, habitat variables were converted into categorical variables equivalent to low, medium, and high values to make it easier to visualize and interpret the results. All right, I'm going to go ahead and move into the results of the analysis. So the first couple of plots are from the VAST model, and here we have the standardized abundance for recruits on the left and adults on the right, and the points include standard errors around the annual values. And there are a couple of big take-homes from these that I'd like to highlight. First is that recruit abundance is pretty variable from year to year, which isn't surprising. That's pretty common for a lot of fish. But in the late 1990s and 2000s, recruit abundance generally was above average, and that those horizontal black lines are the average values for the entire time series. And this indicated that this was sort of a time of a high recruitment phase. The most recent 5 to 8 years have, on the other hand, really not seen much recruitment at all, and the last few years have been pretty dismal and below average. By comparison, for adults, there was a pretty steady increase in abundance following the moratorium in 2001, but the last 8 years have seen a pretty steady decrease, most likely related to the recent recruitment failures and continued loss of fish from the population from various sources. I'm not going to show the full results of the VAST model for distribution, but I do want to highlight some observed changes that were supported by the analysis. For the maps on this slide and the next, any areas that are marked in blue colors indicate the lowest abundances for red porgy, and red indicates areas of highest abundances or hotspots for them, basically. So these maps are specifically for recruits. The top few maps are from 1996 to 2000. The bottom are the more recent years, 2016 through 2019. And these also are equivalent to those high recruitment phases versus low recruitment phases that I mentioned in the last slide. So one of the more striking patterns in spatial distribution of recruits is that the change from recruits being fairly widespread during the high recruitment phase in the late 1990s to a more restricted distribution in the most recent low recruitment phase, with recruits more or less disappearing from the northern and southern ends of the survey and from many of the shallower areas. Recruit distribution also seems to be sort of coalescing into the center of the region as recruitment has decreased. All right, by comparison, the adult distribution really hasn't changed much regardless of changes in abundance. Again, these are equivalent to those two time periods of high abundance for adults, low abundance for adults, top versus bottom, that I mentioned on the initial graphs. And adults are generally found throughout the region and from the mid to outer shelf, with highest abundances in the center of the region. And there was no evidence of any type of shift either north or south for adults, or any evidence of them disappearing from any particular region, regardless of if their abundance was increasing or decreasing. So in addition to defining if there have been changes to the distribution for each life stage, this exercise also allowed us to look at the overlap between recruits and adults. So for example, a couple of examples for recruits on the left, adults on the right. We do see a lot of overlap between the two stages in the center of the region and at moderate to high depths. And this is sort of regardless of if the population was either in a high recruitment or abundance phase, as shown here on the left, versus a low recruitment or abundance phase, shown here on the right. That center off of Georgia, South Carolina seems to be pretty consistent for both life stages. All right, and moving on to the habitat models that we looked at. So for each year sampled, we examined the abundance among the levels of each habitat variable, temperature, biota density, hard substrate, and vertical relief. And for the final objective of defining habitat use for recruits and adults, I'll present them as side-by-side comparisons. Recruit graphs, again, will be on the left, adult graphs on the right. And all graphs are set up so that the dashed line represents the lowest category of each variable, the solid black line the medium, and the gray line the highest values or category. Anything with an asterisk was a significant interaction between that variable and year within the model, meaning that the relationship between habitat and abundance did change over time. For bottom temperature, recruit abundance was pretty variable at the three levels of bottom temperature, and there was no significant interaction. In general, though, when recruitment decreased in the more recent years, those decreases were a little bit more pronounced in high and low temperature areas, although that decrease was really seen at all levels of temperature. Adult red porgy, on the other hand, were distinctly more abundant in warm areas historically, but their numbers have dropped. Oops, I missed an arrow, sorry. But their numbers have dropped in those warm areas, in particular compared to cold, as the abundances have decreased for adults in more recent years. Not on top of my arrow game today. For the second variable we looked at, biota density, recruit abundances historically were highest at stations of either low or medium biota density. However, in the more recent years of low recruitment, all areas see few recruits, regardless of biota density, compared to the earlier high recruitment period. And by comparison, red porgy adults were distinctly more abundant at stations with medium and high biota density in the historic time period compared to low. And that difference didn't really change over time, but rather adults have become less common everywhere as their numbers have become reduced. In the case of hard substrate, red porgy recruits were historically found mostly in areas of low hard substrate coverage. And again, in the more recent low recruitment years, all areas have reduced numbers of recruits relative to the high recruitment time period. And similar to the pattern with biota density for adults, the adult abundance was higher in areas with medium and high hard substrate coverage than low. In this case, though, there was a more pronounced decrease in the in the adult abundance in medium and high areas during the recent time period compared to the previous higher abundance period. And last, for vertical relief, during the high recruitment historical period, recruit abundance was higher in low and medium vertical relief areas relative to high. During the recent low recruitment years, the reduction in recruits was pretty notable in all areas, but very noticeable for those in low and medium vertical relief areas. Adult abundances tended to be higher in lower vertical relief areas relative to medium and high historically, but all levels of vertical relief are now seeing similar and lower abundances of adults. So there have been some changes in the habitat use. So just to go back and summarize what we found so far with red porgy. So based on the Chevron trap survey, red porgy abundance in our region remains fairly low, which is especially worrisome for recruits. The spatial distribution of adults was relatively robust to changes in population size, but by comparison, recruits disappear at the extremes of the region and in shallower water when recruitment is in a low phase. We found no evidence of a northward or any sort of directional shift that may be associated with some sort of environmental change. We found high spatial overlap between recruits and adults, especially in the central portion of the region. Recruits are more common in what I kind of refer to as scrubby areas of low biota, low rocky coverage, but their numbers have decreased even in those areas. But this does suggest that areas with less biota and hard substrate are maybe important to recruits and maybe some sort of preferred habitat for them. By comparison, adults are more common in rocky and warmer areas, so perhaps there is some sort of habitat separation between the two life stages that's not captured by simple mapping alone. And the decreases in recruits and adults in all habitat types also suggest that there isn't an obvious refuge area for this species in the South Atlantic. And in terms of implications for management and recovery of this species, the high spatial overlap between the two life stages may be very problematic with the potential for bycatch mortality of recruits when targeting adults. But probably more work needs to be done on what that potential mortality is. And even habitat-based separation may not help because it would be extremely difficult to tell fishers that level of detail of where to fish and where not to. And in general, if we can't figure out a way to protect recruits and enhance their numbers, then recovery of adult spawners to historical levels will be extremely difficult for this species. Unfortunately, the 2020 SERFS sampling was canceled due to COVID-19 restrictions last year, so we haven't had an update on what recruitment looks like for this species since 2019. However, in 2021, we were able to complete a full survey, and that wrapped up in early October. We are currently ensuring accuracy of the 2021 data and uploading it to our databases to make it ready for some data analysis here pretty soon. We are fully anticipating resuming our annual reports and presentations on species trap abundances. And with those, we generally provide updates on annual lengths. And for some species, and probably for red porgy, we're hoping to have updates on either length or age compositions to provide a check on if we're seeing any new recruitment in 2021. And with that, I am happy to take questions while you all look at some happy snaps of the very many people that have contributed to data collection, in particular field work, aging of many, many red porgy over the years, and funding for this work. And that's it. That's what I have today.
Thank you so much for the presentation, Tracy. And I guess I'll leave it on your slide for a little bit. I think most people are familiar with our process here in order to be recognized and get your questions asked. So if you could, just click on the icon that looks like a turkey if you have a question. If it goes to red, that indicates your hand is raised. I believe almost everyone on the webinar is unmuted right now. So once I call on you, please ask your question. I see one hand raised right now. It's Wilson Laney. Go ahead, Wilson.
Thank you, Chip. So Tracy, that's an interesting distributional change in terms of the recruits, with less recruits abundance on either end of the distribution. One question I always ask when it comes to I've been asking when it comes to red porgy is, you know, what about the other changes that we know have occurred in the ecosystem in terms of other species that may prey on red porgy, specifically red snapper and red lionfish? Do you all plan to take a look at the distribution of those two species as well and see if there's a possible species interaction going on there?
Yeah, that's a great question. We hadn't really talked about lionfish too much, and yeah, we've definitely been batting the idea around of looking at overlap between red snapper and quite a few of our other species that are having recruitment difficulties. But there may be some other players too. Those areas are kind of interesting because, well, one, they are, you know, red snapper are common areas at those ends of the survey, but it's also areas where the shelf really compresses together. So I don't know how much of it is other species interaction versus some sort of habitat limitation at those compressed ends. But yeah, it's definitely something we've batted around. If you all were able to attend Kevin Spanik's red snapper diet seminar a couple of months ago, red porgy was not super common in the red snapper diets, but that was mostly off of South Carolina, North Carolina, and we really haven't had a chance to do that same sort of work off of Florida. So it would be kind of interesting, although the difficulty now is if we don't have any red porgy recruits, maybe they're not showing up in red snapper stomachs because they're simply not there to be eaten.
Thanks, Tracy. Appreciate it.
Okay, Nick Maida has a question.
Thank you, Chip. On the summary slide, maybe I missed this, but in terms of the spatial overlap, I thought I saw there's a bullet that says there's high overlap between the recruits and the adults, but their habitat preferences were different, right? The adults like deeper. Go ahead.
Yeah, that's a great question. Yeah, so I think if you just do simple mapping, they like to be, they're both really common in the center of the region, but I think within that there's separation between the two. That's just not captured by those really sort of gross overview spatial distribution maps.
Understood. Thank you.
Any other questions? Jimmy Hull?
Yeah, thank you, Chip. Yeah, very interesting. Tracy, my question is, in the traps that are pulled that show a catch of red porgy, is the composition of the catch, is there maybe some predators like red snapper that are being caught at the same time as the red porgies that are being sampled? Does it show that that is a possibility because there's snapper being caught at the same time?
Yeah, that's a good question. I think the short answer is yes, it's a possibility. The red snapper do tend to be a little bit shallower, or at least, you know, in the last couple of years, although they are becoming more common sort of everywhere compared to red porgy, especially the adults. But in terms of, well, the red porgy that are sort of eating size that would fit in the mouth gape of the red snapper, that is a great question. It's definitely something that we could look at in terms of just isolating those smaller fish and their overlap with red snapper that would also be large enough to eat them.
Right, and then the other thing I would note is that off of northeast Florida, the southern end, where you show a decline in both recruits and adults, is really a hot spot for red snapper population. And
Yep,
also, it seems like, I believe, off North Carolina is also a hot spot. So I don't know, it just kind of leads to that, to somewhat, you could assume that, I think. Thank you.
Yeah, and I think one of the big things we probably need to look at is what sort of competition is happening between the two, even if they are not, it's not a predator prey or not exclusively predator prey, because Kevin's definitely the diet specialist, but I have some vague recollections that they both tend to slurp things up from the bottom. Especially depending on what their sizes are. So it's very possible that they're competing, at least in some of their life stages, for some of the benthic prey that they both have shown some interest in eating.
Yeah, and as well as habitat. I can tell you that off of Ponce Inlet, we used to be able to catch a lot of black sea bass. And now all of the bottom, which for sea bass generally is low relief, as you described there. Yep. And they're gone. And all you'll catch now is snapper of all sizes, the smallest to the largest. So the habitat's definitely been taken over also. Thank you a lot.
Yeah, thank you for that question, Jimmy. And just to build on a bit of what Tracy was talking about, we did have a EcoPath model with EcoPath with EcoZim presented to our SSC 2 weeks ago. And it was talking about the competition or potential competition between red snapper and red porgy. And needless to say, it's based on some of this data that Tracy presented here today. But it indicated that there was very little competition, very little diet overlap between the two. But I think that does need to be investigated more, like Jimmy says and Tracy has said. And if you want to find out more about it, Lauren Gentry is going to be giving a presentation on that at our council meeting coming up in December. Does anyone else have a question? If not, I will ask a question. All right, I'm not seeing any hands. And if you don't feel comfortable talking in the on the webinar, you can always type it into the question box as well. Tracy, I'm wondering if the spawning season is varying over this time period. Is there potential of shortening of the spawning season, or which could be causing some of the changes in recruitment?
Oh, that is a great question, and I really don't know. Yeah, so I know Dave has found that the age and the size at 50% maturity does tend to shift, or has over the 30+ years. But I'm not sure we've ever had good enough data to look at the spawning season itself. They're winter spawners, and because we're using a fixed gear, we really can't put anything in the water before April 15th. And then our sampling season ends October 15th. So we are missing quite a bit of the spawning season to be able to address that. Yeah, Wally, I don't know if, I think Wally's on the call. I'm not sure if he remembers anything on if we've had any extra winter efforts to look at that a little bit more comprehensively. But that's one of the big limitations right now, is we just haven't had a chance. We just don't have the samples historically to really look at that very well. But it is very possible.
Go ahead, Wally.
Yeah, I'd just say I think Tracy covered that very well. Because of the spawning season being winter months and us not having gear in the water, we've gotten samples from the winter occasionally, but nothing consistent at least with the traditional chevron trap sampling.
Right. So Dave, if you want to go ahead and speak, you should be unmuted. His response to the question says yes, can't assess based on our sampling season. And Dave Wyansky works with South Carolina DNR doing a lot of the reproductive work. Any other questions? Frank, I see you have your hand raised.
Thanks, Chip. Thanks, Tracy. I was wondering if I could get a copy of the reprint of the paper you just put out.
Yeah, absolutely.
Thanks.
So what is the smallest fish you're seeing when you're saying you're observing some of the H0 fish? How small are these fish?
Oh, you're gonna make me remember what the trap mesh dimensions are. Bigger than that. Yeah, it's palm sized is about the smallest ones that we tend to retain in the traps, depending on whose palm it is. And I want to say Marcel mentioned that we did see some small ones this year when he was out sampling, but we haven't finished up the data with all of the cruises just yet. So hopefully we'll have some good signs this year. But yeah, it's a little bit bigger than palm size, hand size.
Thank you. It's a good visual for me to know how big it is.
I can't convert between centimeters to inches very well.
Wilson Laney has his hand raised.
Yeah, thanks, Chip. So your question prompts me to ask Tracy is whether or not they have any issues with predation inside the trap. You know, is there a chance that the recruits could be consumed by larger fish inside the trap? Is that an issue?
We have definitely seen gag and red snapper eat tomtate in the traps, and maybe some black sea bass. I'm not sure we've ever gotten any good footage of any porgy getting eaten. For a couple of years, we had GoPros suspended inside the trap and looking back towards the mouth. So we have some evidence of in-trap predation. But despite several hundred of those videos, it's very infrequent.
All right, thank you for that. And Wally responded on the question of the smallest, and it appears it's about a 6-inch fish total length, 156 millimeters.
Yeah, that's Wally's hand. That's smaller than Wally's hand.
Any other questions? I am not seeing any other questions. So with that, I want to thank you for the presentation today, Tracy, and all of your partners. You know, I think we learned a lot about red porgy habitat distribution. And can you describe the video we're seeing now?
These are our 2021 highlight reels, and I'm just using them to distract you all.
Well, thank you. And yeah, there's a common sight right there with a greater amberjack.
And the lack of red porgy, unfortunately.
All right. Well, thanks everyone for coming today. And we're going to have another presentation in January. We're working on setting that up. If you have any requests or any presentations you would like to see, please don't hesitate to contact me. My email is chip.collier@safmc.net. Or you can also just go to our website and contact anyone within the South Atlantic Council, and we're happy to work with you to try to get something established. Thank you guys for coming today, and look forward to seeing you guys soon.
Thank
care.
you.
Thank you, Tracy. Thank you, Chip.
Thank you, Tracy. Thanks, Chip.
You want me to stop sharing?
I'm getting ready to end the webinar, so it will.
We'll just take it away.